Hey guys and gals, we are going to have a discussion today on the shape of the cell and the surface of the shell. So we're talking about cell shapes and the cell surface. So let's dive in. While we're talking about cells, it's worth reviewing cell theory. I know I emphasized this earlier on in this course last week, but I need to emphasize this again. It's bolded, it's underlined. The cell is the simplest structural and functional unit of life. That is the smallest living thing. If it's smaller than the cell, it is not alive. So when we think of cell theory, all living things come from cells or cell byproducts. When we look at cells, every cell in the body comes from a pre-existing cell. The only thing that can make another cell is the parent cell. And of course, obviously, if you sit down and think about this, this will beg the question, well, what about the first cell? Where'd the first parent cell come from? Something had to make the first cell. And I don't have a good answer to that question. I'm sorry. That's beyond the scope of this course. When we look at cells, all cells that are similar to each other will have similar biochemical characteristics. And even if the cells are wildly different from each other, like a red blood cell to a skeletal muscle cell to maybe a bull sperm cell, you may have used those in lab this week. As we're looking at even wildly different cells within an organism, they're still going to have similar biochemical, there's similarities or characteristics. They still will have the same general kinds of chemical reactions in them that keep them alive. Now, when we look at cells, there's about 200 different kinds of cells within human beings. And again, this is human anatomy, physiology, number 1, Bio 214. And in this class, we're going to focus primarily on human beings. No, I am not going to ask you to memorize the 200-ish different kinds of cells, especially in this PowerPoint presentation. No. But it's worth noting that there are lots of different kinds of cell shapes. Let's talk about some broad categories. We have squamous, cuboidal, and columnar. When we think of a squamous cell, I always think of squashed squamous. They're like little pancakes. So here's some squamous cells on the bottom of the screen. Boy, do I really wish I had a bigger mouse on my computer. I'm sorry about that. I guess I just don't. And when we look at these squamous cells, guys, gals, they're thin, flat, scaly. I like to think of them as lots of little pancakes that can be either a single layer of pancakes or multiple stacked layers of pancakes. Up next, we have cuboidal. They're kind of squarish looking in a microscope slide, but in 3 dimensions, they'd be, well, shaped like a cube, hence the name cuboidal. And those cuboidal cells are about as tall as they are wide. And then we have columnar cells. And as we look at columnar cells are, well, for lack of a better term, taller than they are wide. They're column-shaped cells. Now, these figures don't necessarily do it justice because these figures that we have from your textbook really have 90-degree corners on the cells. And in real life, the corners are generally speaking a little bit more rounded to each other, but especially while we're talking about it in a lecture, we're going to keep the pictures and the figures really clear, really crystal clear and obvious for you. Now, it's worth emphasizing, guys, gals, that as you slice through the cell to make your microscope slides and to look at the cell, depending on how you slice the cell, it's going to look different. If you have a longitudinal slice versus a cross-sectional slice or transverse slice, you might get a large oval, you might get a small circle, or you might get something that's in between, even if you're slicing through the exact same cell. Some other cell shapes that we have that are fairly common within the human being are polygonal. Polygonal is kind of the miscellaneous category. If they're all a bunch of cells that are irregularly shaped, we call those polygonal shapes. Stellate cells are shaped like stars, hence the name stellate. We could also have spheroidal or ovoidal cells. Pictured here for the stellate cells are some neurons, some multipolar neurons. Pictured over here with the spheroidal cells are some leukocytes or white blood cells of the body. And depending on the leukocyte, particularly if it's a phagocytotically active leukocyte, it's going to change shape as it morphs and makes pseudopods and phagocytizes or swallows the bad guys. But generally speaking, spheroidal is a good category for the leukocytes. We could have discoid shells. Here are some red blood cells, also known as erythrocytes. And as we look at these discoid cells, one way that this has been described is a biconcave shaped cell. The reason these cells are discoid is because most of their organelles have been removed from the cytoplasm. Notably, there is no nucleus in these discoid-shaped cells. I always like to think of these guys as jelly-filled donuts. I mean, really, if we could reduce everything in life to some sort of a donut analogy, that would just really make me very happy. We also have fusiform cells or spindle-shaped cells, and as we look at these cells, they're nice and thick in the middle and pointy at both ends. As we look at this, this is a good representation of smooth muscle cells. And then we can also have fibrous cells. Notice as we look at these fibrous cells, they're much longer than they are wide. And when cells are this long, they need to have multiple nuclei present in them. And these multiple nuclei present in them help to control one little area of the cell. So as we look at these cells, this fibrous cell is going to be a skeletal muscle fiber. Now, as we look at human cell size, they're about 10 to 15 microns or micrometers in length. Recall during lab when we talked about this little Greek lowercase m right here, that's the Greek letter mu or lowercase m. In a medical setting, a common abbreviation that's being used that's easier with your keyboard is MC for micro as opposed to mu for micro. So just make sure that you're consistent with whatever you use. As we look at human cells, this is the average. There's a lot of variation. Think back to lab when you're looking at the sperm cell, the red blood cell, the squamous cell, and then you also looked at the skeletal muscle cells. Even though those were all representative cells that are present in human beings, those cells were wildly different from each other. A human egg cell, for example, is the large end of the spectrum for human cells. It's 100 microns in diameter. If you have awesome eyesight, you might just barely be able to see a human egg cell with your naked eye. Some nerve cells, even though you can't necessarily see them because they're so skinny, they can be over 1 meter in length, especially when you look at how the axon of that nerve cell. Now, as we look at cells in the human body, cells can't be too big. There are some upper thresholds, some upper limits on the size of the human cells. If the cells get too big, we have issues with membrane transport, diffusion of nutrients within the cytoplasm, or just the membrane itself losing integrity and rupturing. Think of it like a big bubble. It's the bigger the bubble is, the more likely the bubble is to pop. So as we look at these cells, there's a Goldilocks zone, not too big, not too small. It's the just right zone. And this just right zone is where we have the correct proportion of volume to surface area. And what we find, if you think way back to your geometry class from when you were in high school, 4/3 pi r cubed is the equation for the volume of a sphere. So that 4/3 pi r cubed, pi radius cubed, as the radius increases, the volume is going to increase with a cube root. So it gets really quick, really large, really quickly. And that's going to be faster than surface area. Surface area is going to be based off of a squared function compared to a cube function. So the volume increases faster than the surface area. And as cells get larger, the difference between the surface area and the volume is magnified because of some of the basic geometric math involved. So as we're studying cells, there are 3 classic microscopy techniques that we use to study the cell. Here in Bio 214, we're going to spend a ton of time using visible light microscopes. And I know for all of my lab sections, I talked about how your visible light microscopes are some of the best visible light microscopes you are ever going to get to use in an undergraduate setting. So I just need to give a shout out again. UW-Eau Claire gives you fantastic microscopes to work with, particularly for an undergraduate level class. And here we have a eosinophil. Notably, it's one of the leukocytes that's stained pink or eosin pink. And this is a typical light microscope image, and this is definitely going to have been, or this image is definitely going to be at 1000x using oil immersion. We can also look at cells using transmission electron microscopy. The nice thing, here we go. The nice thing about transmission electron microscopy, or TEM, is that you can see a lot of interior detail. You can transmit information from the inside of the cell. So as we're looking around here, we can see different organelles. There's the nucleus that's very large, and you can make other inclusions and organelles within the cell itself. So hey, that's pretty cool. And you can zoom in even closer. But the gold standard for those super cool images that you see are the scanning electron microscopes. And at UW-Eau Claire, we are super lucky. We actually have an electron microscopy lab that if you decide to take some upper level biology classes or graduate level biology classes, you could spend some time working with that electron microscope. It's a fabulous opportunity. I highly recommend it for you. Scanning electron microscopes scan the surface, and they give you fantastic resolution, but only of those surface features. So here in the lower right, I have a SEM image of a cell from a human being. Now, as we're looking at cells, all cells are going to have the same basic components. There's the outer barrier. That outer barrier is called the cell membrane, aka the plasma membrane. It separates the inside from the outside. It separates the it separates the extracellular environment from the intracellular environment. And this plasma membrane, this cell membrane, is going to be made overwhelmingly of proteins and lipids. And the lipid, if you think back to when we talked about biochemistry and we had our chemistry review section, the lipid that's most common in the cell membrane is going to be a phospholipid. Now, the interior of the cell, the inside of the cell membrane, is called the cytoplasm. And within the cytoplasm, we have all of the organelles, we have the cytoskeleton, we have inclusions, and the cytosol itself. One of the organelles, at least according to your lecture textbook, this edition of your lecture textbook, the nucleus is categorized as an organelle. Depending on the textbook and the publisher and the edition, sometimes the nucleus is not considered an organelle, sometimes it is. For our lecture class, with our lecture textbook, we are considering the nucleus an organelle. The cytoskeleton, we'll talk about in more detail later on, I believe in our next discussion. Not this recording, but 2 recordings from now, we'll talk about the cytoskeleton. We also have, just briefly though, when we think of the cytoskeleton is a bunch of filaments that support the cell and the organelles inside of the cell. Inclusions are nutrients or particles that are being stored for future use. And then we have the cytosol. The cytosol you can think of as the liquid on the inside of the cell, the intracellular fluid, ICF. That liquid on the outside of the cell, the extracellular fluid, can also be categorized as interstitial fluid. It's the fluid in between the cells. And sometimes you'll hear me call it the extracellular fluid. Sometimes I call it the interstitial fluid. Please note, those are synonyms with each other. This is a really good thing to write in the margin notes if you're following along with the printed out PowerPoint. Extracellular fluid is a synonym with interstitial fluid. Here's a diagram of the cell. This is one from the textbook that you're using. Note that this label diagram of the cell is different from the label diagram of the cell that you have in your lab manual. So you have the same diagram 2 different ways. I'll spend some time highlighting some of the key structures here. Here we have the nucleus. On the outside of the nucleus, we have the nuclear membrane. In the center of the nucleus, we have the nucleolus, or nuclei if there's plurals. And continuous with the nuclear membrane, right next to the nucleus, we have the endoplasmic reticulum. If there are speckles on it, those speckles represent ribosomes, and the speckled endoplasmic reticulum is called the rough endoplasmic reticulum. The non-speckled version right here is called the smooth endoplasmic reticulum. We have another stack of membranes right here that's distinct, not connected or not continuous with the membranes of the nuclear envelope. This stack of membranes right here is referred to as the Golgi apparatus, and generally speaking what ends up happening is that things are packaged up in the rough ER, sent to the Golgi apparatus, processed, repackaged, and then exocytized, secreted into the extracellular environment. Notice how the cell membrane up here folds up and down. It's folding up and down to increase the surface area. We call those little folds microvilli. And then here, connecting one cell to another cell, we have a cell-cell junction known as a desmosome. Here we have an inclusion filled with triglycerides. We call this a fat droplet or just a storage vesicle, more generally speaking. This region right here is known as the centrosome, and that centrosome is made of individual centrioles. Usually we'll have 2 centrioles per centrosome. Coming out of the centrosome are going to be mitotic spindles, or more broadly speaking, the cytoskeleton. You can see that there's some bean-shaped organelles that have a bunch of little folds on the inside. These bean-shaped organelles are called mitochondria. We also are going to have a little gap in between the cells. This gap in between the cells is the interstitial space. But notice how the plasma membrane or cell membrane over the middle cell butts directly up or right next to the cell membrane that's right next to it. There's just a little tiny intercellular space in between these guys. Notice how we have most of the ribosomes shown here. Most of the little purple dots are stuck to the ER, the rough ER, but we also can have free-floating ribosomes within the cell as well. So this was just a brief overview of the cell. We have a separate lecture period completely dedicated to talking about the organelles, their structures, and their functions. So we're going to focus more on them at that time. Right now we need to focus on the plasma membrane. That plasma membrane is the outer environment, the outer border of the cell. It separates the exterior environment from the interior environment of the cell. A synonym for the plasma membrane is the cell membrane. If you were to look at the plasma membrane using a TEM, transmission electron microscopy, you would see the plasma membrane as a darkened line, 2 dark parallel lines that are right next to each other. Those 2 dark parallel lines represent the phospholipid bilayer, the 2 layers of phospholipids that make up the plasma membrane. Something that the plasma membrane does is it controls what goes in and out of the cell. It's like the bouncer at the nightclub. It will look at molecules that want to get into the club, and it'll give them a thumbs up or the thumbs down. And on the same token, if something doesn't belong in the cell any longer, that plasma membrane helps that, whatever that thing is, whatever that molecule is, the plasma membrane will help get it out of the cell. So the plasma membrane, practically speaking, is going to allow needed nutrients to gain access to the cell and help get rid of waste nutrients or waste byproducts from the cell. Most of the plasma membrane is going to be lipids or fats, particularly phospholipids. And if we look at it in terms of the number of molecules, overwhelmingly, the number of molecules present in the plasma membrane are going to be lipids. But we have a caveat here. Those phospholipids that make up the plasma membrane are really small, so they're high in number, but they're low in mass. And of the lipids that make up the plasma membrane, phospholipids are by far the most common. Recall back to when we talked about biochemistry and the chemistry of life. Phospholipids are amphipathic. What does amphipathic mean? If you forgot, write this in the margin right now. Amphipathic means there's a hydrophobic and a hydrophilic region on the phospholipid. They like to mix with water and they don't like to mix with water. The head of the phospholipid, where the phosphate and the amine groups are, like to face towards the water. They're polarized, polar groups that mix well when exposed to water. And then the fatty acid tails of the phospholipids, those do not like to be exposed to water. So those are non-polar functional groups that like to be buried within the middle of the phospholipid bilayer. These phospholipids bounce around within the cell membrane. Sometimes this is referred to as a fluid mosaic model, the idea that all these individual parts that make up the plasma membrane look kind of like the squares in a mosaic picture, but they're bouncing around, they're fluid, they're constantly in motion. So here we can see a phospholipid where we have the polar head and the 2 fatty acid tails. We also in our plasma membrane have cholesterol molecules embedded in the plasma membrane. When we talked about the chemistry of life, we had mentioned how cholesterol molecules add stiffness or rigidity to the plasma membrane. And as we're looking at this plasma membrane, there's mostly phospholipids. That's the most common molecule. But the proteins, those big purple proteins that are present in the plasma membrane, are very large. Some of the proteins go all the way through. We call those transmembrane proteins. Some go all the way through and have a hole in them. We call those pores or channel proteins. Some proteins are only on the surface, one side or the other side. So if it's only on the inside or only on the outside, that's a peripheral protein. And then we also are going to have proteins with carbohydrates on them. Glyco for glycogen, the animal carbohydrate, and protein because, well, the glycogen is attached to the protein. So a glycoprotein is a protein with a glycogen molecule on it. We could also have a glycolipid, a glycogen attached to a phospholipid within the cell membrane. And just to give us a frame of reference, we have intracellular on the bottom of the screen and extracellular on the top side of the screen. So the cholesterol is a minority of the membrane lipids. And just to review briefly again, cholesterol stiffens the membrane. The more cholesterol you have, the stiffer your membrane is going to be. When we look at glycolipids, a lipid, phospholipid with a glycogen molecule attached to it, so a lipid plus a carbohydrate attached to it, it's a minority of the lipids. And when we look at these glycolipids, they help contribute to something known as the glycocalyx, a coating of carbohydrates on the surface of the cell that serves to help to identify the cell within the body. And I put a note there to stop recording because that's the halfway point for this lecture discussion. So I'm going to go ahead and stop our recording. But before I do, guys, girls, I want to emphasize that if you have any questions, any comments, or any concerns, please feel free to email me or to swing by my office hours. And as always, happy studies.